Welcome back, students. This is the concluding video on the topic semi-conservative replication. Those who have not viewed the first part, A part, I suggest you first go through that and then come back. Otherwise, you may not be clear with certain concepts. In my previous lecture, we have discussed the very accepted hypothesis that replication of DNA occur in semi-conservative manner, and also Meselson and Stahl's experiment, a very wonderful experiment to test this hypothesis laid by Watson and Crick. Let's recap the experiment fast. Meselson and Stahl started their experiment with E. coli cells cultured in N15 for several generations so as to start the experiment with cells having only heavy isotope of nitrogen N15 in their molecules, including the DNA. DNA has purines and pyrimidines bases, which is rich in nitrogen, and our molecule of interest is DNA. So such cells were harvested from the N15 media, and they were transferred now to a lighter isotope. Source of that nitrogen with light isotope was NH4Cl. This can be an important question. What is the source of nitrogen for the experiment? To have the light isotope, it is NH4Cl. Yeah, here you have to write 14. What was the source of nitrogen before starting the experiment to have all the cells with heavy isotope? That was NH4Cl with isotope 15. Okay, so they transferred the E. coli cells to N14. So their experiment starts here with E. coli cells cultured in NH4Cl with lighter isotope of nitrogen. What is heavy and light means? See, these are not radioactive isotopes. Heavy isotope means when these DNA samples would be subjected to density gradient centrifugation, the heavy isotope, that is the N15 one, incorporated DNA strands would settle at the bottom of the spinning vial. The gradients are formed because of cesium chloride solution. Cesium salts being heavy, they settle at the bottom. They dissociate in the solution and they settle at the bottom, forming a denser gradient. Some of the cesium salts may diffuse back, form a slightly lighter gradient. So that would be another zone. And a very few atoms would be at the topmost layer. So that forms the lightest gradient. So that likewise cesium salts form 3 different gradients. So accordingly, the DNA, the heavy one will come in equilibrium with the heavy zone, the lighter one with the lighter zone, that is N15 in the deeper zone, and the N15 in the lighter zone. And the intermediate zones would be occupied by such DNA duplexes which have both N15 and N14 incorporated. So they form a intermediate band like. DNA would be visible in the form of bands. Bands are nothing but DNA. We cannot view DNA as such in the vial. They will appear in the form of bands. So that is the whole thing about centrifugation. High-speed centrifugation is done. Now, these cells from N14 medium were isolated. That means they were harvested. DNA was isolated and subjected to centrifugation. So what was the analysis? Meselson and Stahl started performing centrifugation from 0 generation, and we know 0 generation is the start of the experiment. That means cells which are having completely N15 in them. Okay, they have not started now incorporating N14, so they are definitely showing bands at the bottommost gradient. 100% heavy bands would be seen, indicating that both the strands of the DNA duplex are having still N15. Then they gave certain time, that is E. coli doubling time is 20 minutes. So after every 20 minutes, starting from the 0 generation, every 20 minutes they started analyzing DNA samples, subjecting it to centrifugation. So the first generation, that means after those cells, the initial cells were allowed to divide, when DNA samples were extracted and analyzed through centrifugation, they found that they formed bands in a gradient in between the heavy gradient and the lighter gradient. Plus indicates heavy and minus indicates less gradient, lighter gradient. So it was in an intermediate region where the bands were seen. So that indicated that those DNA may be definitely having both the types of nitrogen incorporated in each strand, N15 in one strand and N14, the lighter one, in the other strand. So that suggests that the parental strands of the original cells would have separated and incorporated the newly given food nitrogen from the newly given medium on it. That is why these two types of strands, and that is why the intermediate band formed, indicating that both strands would have been in there. Then waiting for another 20 minutes, they again analyzed DNA samples. That means from starting time 40 minutes. So the next generation, second generation, that is at 40 minutes. So they found that the bands were in two regions. Here 100% heavy, here no heavy, 100% intermediate, because the parental strands are separated, so no pure both parent strands would be there. So 100% intermediate, hybrid DNA. Now they found that there were bands equally, two types, intermediate band and one band also on the lighter zone. So again, that was a suggestion, an indication that again these intermediate hybrid DNA samples, or the two strands, has separated and incorporated new strands, because of which such two bands were visible. That means the heavy N15 strand would have incorporated light, of course, in as many number of DNA molecules, and the lighter bands would have again incorporated lighter band only. So two types of bands were visible, 50-50%. Gradually, further generations samples were analyzed, and it was found that the intermediate bands became fainter and fainter. No heavy bands were seen at all, and the lighter bands were predominant, indicating that only N14 was being incorporated in the newly formed strands. See, now this is the question I left in the previous video for you to do, and as given in NCERT, I'm sure most of you would have separated out strands like that to calculate the proportion and all. But for practice it is okay, but think during examination, you have to save your time. So the easiest way of calculation is, you need not work out each strand separated out and then addition of new strands. So whatever is a generation asked, like after 80 minutes here, it could be more than 80 minutes asked, you will spend a lot of time doing this work. So just get the concept clear that at 0 minute it is 100% heavy. That means both the strands of the duplex is having N15. So we are following the semi-conservative mode. So what happens? The two strands separate. Intermediate ones, because both strands incorporated in the lighter isotope strand. 100% hybrid, till here it is okay. The next generation, that is after 40 minutes if asked, very simple. The intermediate duplexes would separate each of the parental strand, and the heavy ones will incorporate lighter, and the lighter ones will also incorporate lighter. So what you do is, instead of drawing out all this, you start with the two heavy strands throughout the generation time, throughout the experiment, whatever generation time given would be only incorporated once in any two duplexes, because they are only one set of duplexes. So ratio we have to see. So you separate out and you go multiple, every generation you take a multiple. For example, here one duplex. So 1 into 2, 2 duplexes, 100% hybrid. 2 into 2, 4 duplexes. Out of 4, 2 intermediate, definitely, because there were only 2 duplexes with heavy strand, and 2 lighter, totally lighter. So 50% hybrid, 50% light. That means if you are asked ratio, 2 out of 4 is hybrid, 2 out of 4 is that is duplex, is light. So 1:1 is the ratio. Okay. Next 20 minutes, that is 60 minutes, the generation will give you 4 into 2, 8 duplexes, out of which only 2 will be hybrid, because we are starting with only 2 heavy strands separated out. So 2 out of 8 is hybrid, so remaining is 6 out of 8 would be light. Then again next generation, you make 8 into 2, 16. 16 duplexes, you need not work out all this. 16 duplexes, out of which only 2 would be hybrid, remaining 14 would be light. So 14:2 or 7:1 is your answer for this question. You can even work out further. 100 minutes, 16 duplexes into 2, 32 duplexes, out of which 2 will be only hybrid. Every time only 2 will be hybrid. And the remaining would be light. So 15:1 would be the answer for 100. So this is for 80 minutes. So you bother about this one now, but learn to work out how to work out with this calculation. This will be easier. I hope that is clear. And light and hybrid is asked, so always mention light first. If you mention 2:30 and leave, the answer may be wrong. Otherwise, you should mention 2 hybrid is to light, 30 light, like that. So be careful on that. Experiment conducted by Taylor and his associates. Taylor et al. Means his associates. Is also mentioned in NCERT. So we'll go through it in brief as given there. He performed experiment on broad bean. So you should remember the name of the plant, Vicia faba, scientific name. They tagged DNA of this root tip cells with radioactive thymidine, H3. They cultured the cells in this radioactive thymidine, nutrient with radioactive thymidine, and they started their experiment with cells having DNA or the chromosome incorporated with radioactive thymidine. Then they cultured the cells in normal thymidine, normal medium, that means medium with normal thymidine. And they found that first generation, the metaphase chromatids were showing one arm unlabeled, not radioactive, and the other chromatid in the metaphase stage radioactive. So that also proves semi-conservative nature. Second generation metaphase chromatids appear 50% having one arm each radioactive and the other 50% totally unlabeled. So this also proved the semi-conservative mode of chromosome replication. Of course, if chromosome is duplicated in semi-conservative manner, of course DNA also has replicated in semi-conservative manner. That's all about the semi-conservative nature of DNA replication. But I would like to remind you that this is the model how DNA replicates. That means the two strands separate and how the parental strands incorporate the new strand. But how does replication occur? There are so many enzymes involved. There are various mechanisms of replication. So how one strand replicates, how the other strand is replicating in an entirely different manner, these things we will cover in the next video. We will learn about semi-discontinuous mechanism of DNA replication. That is, one strand replicates continuously and the other new strand replicates in fragments. So that is there. But however the mechanism is, every time the new duplexes always have one parental strand and one newly synthesized strand. So that nature is semi-conservative. Mechanism we will come to in the next video. Thank you for watching. See you in the next video. Until then, take care.